Hello, hello, everyone. Welcome to the Soil Food Web School four-part webinar series, The Science of Soil Fertility in 3 Steps. We're so thrilled to have you here joining us. Thank you for being here. I know everyone is going to start to come in now. Please start putting your name, where you're coming from, so we get a feeling where all the parts of the world we are. My name is Isadora Schmidt. I'm here covering for Nick. Ha ha ha. I'll be your host today. And this is the first of our webinar series, Discover the Beneficial Soil Microbes Around You. Oh, there you go. So this series is designed to help you unlock this incredible power that the living soil has. It's not just to understand what they do, who they are, but it's also to know how we can regenerate landscapes, how we can grow more nutrient-dense food, and how we can reduce the dependence of chemical inputs, okay? So whether you are a grower, whether you are a consultant, a researcher, or just a micro geek like us, or just curious about regenerative agriculture, this session is going to give you a deeper appreciation for the biological processes. And yes, please say hello again. So tell me where you're coming from, and we'll get started. So I'm really, it's such a pleasure for me to be here with my fellow Soil Food Web mentors and colleagues and friends. And please introduce yourself, Carla.
Hi, everyone. You saw me before. I have the honor to be the science leader for the school, also instructor, and I see a lot of my students here. Thank you for joining us. It's always a pleasure to have you guys here. I'm with the school for almost 6 years now and have the honor to learn from the source, Dr. Elaine herself, and also have my consultancy company called Soil Domains, and we work in Oregon and Brazil. I'm a biologist by background with environmental science, environmental engineer master's and environmental science PhD. And work here at the school is just a great honor. Gerald?
Hello, everyone. Well, as you are seeing there, my name is Gerald Ramirez. I come from the beautiful Costa Rica in Central America. So if there is any Latin American person here, please raise your hand and let me know that you are part of this great movement today. I am an instructor for the Soil Food Web. I've been with the school for 4 years and a half now. It's been an incredible journey of teaching and learning at the same time about soil restoration. Um, I am an agronomist. My interest in all this started because I feel like the current chemical approach that we all live in is becoming unsustainable. So we need to look for new and different ways of doing things. And I want to do something different. I want to impact the world in a different way. That's why I've been here. It's exciting to be part of all this, and it's exciting to have you guys here from all different places in the world. Apart from working with the Soil Food Web, I currently have a project with a place called Armonia in my country, where we are developing an organic waste management strategy to deal with their food scraps and all the waste they produce to convert it into really good compost that hosts the microbial life that we are going to talk about. And yeah, that's what I have for now. Really excited to be here and hope to give you guys some really good ideas for you to continue with your project.
That is so cool. Look, I'm looking at all that. Bridget is here. She was in our workshop in England. She's saying hi to you, Gerald. We have people from all over the world, from California, from Florida, from Canada, from the Netherlands, from the UK, Norway. It's super cool, really nice. And I'm Isadora. I live in Spain. I'm a soil consultant here. I'm also a soil food web mentor. I just like my colleagues, I really passionate about this. I am known as the crazy microbe lady here in my area because it's just a lot of fun, right? And it's a lot of fun to work with farmers and food producers as well. This is one of the things that I appreciate. I also collaborate with agricultural schools. I do a little bit of research. And it's really happy to be here. Really excited. What else do we have for today? Okay, so This is a 4-part webinar series. The first one, we're going to start talking about the microbes, right? We'll talk who they are. We're going to see what they're doing. And then for the second webinar, which is going to be next week on the 17th, we're going to talk about how we actually multiply them. So this is about composting. It's going to be with Dora, with Nick, with Brian, and myself. And we're going to be talking about large scale and small scale composting, how we multiply it. And then their third webinar, after we learn about the microbes, we know how to get them there, we are going to see how we actually apply them in soil. We're going to see real life cases. Adam York is going to be there with us. He has 10,000 hectares of corn and soil in Illinois. I cannot imagine what that feels like. 10,000 hectares. And he also has a separate business where he's supporting his neighbor farmers with biological extracts. So I think it's going to be super, super exciting. Stay tuned. And last but not least, the fourth part of this is going to be your community house party. It's not the first one. We've done some before. It's a lot of fun. Anyone can connect. We'll have different breakout rooms with different topics, and we'll be just, having fun. Do you have anything to say, Carla? Did you enjoy the last
Oh,
party?
it's always a lot of fun, like you said, and we don't see the time pass. It's just like we can stay days actually. And I think the most precious part of the community house party is that we can hear you guys. We can harness our community and learn from each other on a sense that we really open up our hearts and how soil science help us change one form at a time. What about you, Gerald?
Well, I wasn't in the last soil party, but I heard it was awesome. And I'm sure that it's a great opportunity to share insights and share different experience. Every time we build community in any of the spaces that the school creates is such a positive energy and such a beautiful exchange of, experiences and thoughts. So I think it's great if you guys can make it to the community house party.
Yeah, because we
100%.
can't do this on our own, right? We need support
That's
too, just like farmers.
Mhm.
They're not supposed to know everything. We're not
Absolutely.
supposed to know everything. And
Hmm.
and leaning on each other and understanding, seeing who's, like we're vibrating in the same frequency just really helps. And yeah, so
Mhm. Yeah.
yes, exciting. All right, let's move on. So today's webinar, Discover of the Beneficial Soil Microbes Around You. So we're going to start with Carla, with Dr. Carla Portugal,
Carla.
talking about, sorry, to embarrass you a little bit, identifying beneficial microbes. We're going to see how wonderful they look. And right after that, Gerald is going to start talking to us about how do we actually work local, because there's a lot of bugs in a jug now. There's a lot of people trying to, because we understand the power of biology. We've seen in the last years how powerful one microbe can be. We have rhizobium seeds, we have trichoderma, but when we work with the entire soil food web, it's just magical. So yes, we're going to talk about that. And right after, we're going to follow by a live Q&A session. So make sure to write your questions if you're in YouTube. You can write your questions in the chat. If you are here in Zoom, please write it. There's a Q&A area chat box that you can write, okay? And we'll get them at the end. And we will try to address them as much as we can. Okay, so instructions, basic instructions. So we're not all, we need limits. You will all be muted for the duration of the webinar, okay? Enter your questions in the Q&A and feel free to talk with anyone else in the chat and connect with other people, okay? This is the time to do it. So without further ado, I hand it over to you, Carla.
Thank you, Isadora. Thank you, everyone. I have to start telling you guys that I'm a super nerd when we start to see those little amazing creatures on the microscope. So you might hear me to say a lot that they're cute, they're cute. And like everyone jokes with me, there is any of them that are not cute? No, there's not. We just need to better understand those guys and how they can help us recover soils everywhere. So let's talk a little bit about identifying beneficial microbes or microorganisms. As you guys remember from Dr. Elaine explained the soil food web, and I will do my best to get close of her versatility and how we fall in love about soil food web with Dr. Elaine. The most important bigger picture that we cannot forget is that we need to understand the system as a whole, as a unique complete scenario where each player has distinct roles depending on which angle we are analyzing that web. So it's a very intricate and complex net that Dr. Elaine just do her magic and make us fully understand the soil food web as a whole. I think I can use the word microcosmos because, in fact, that it is what it is, right? We have the whole set of aggregates that those little guys will do the magic and transform the nutrients that are there on a form plants cannot absorb to a format that plants can easily take in and grow in a more healthy manner. So when we talk about soil regeneration, we cannot leave soil food web behind. There is no way to not talk about the entire system without all the parts. We know by fact that universities talk about the physics, the chemistry related to soil and the structure, but it's only during recent years that some of them are starting to include the biology portion of that. And we cannot blame anyone because this is how the educational system was built. We learn things in separate boxes. And what we do here at the school is help us rethink how we connect all the knowledge we have and interconnect all those pieces. So having the biology or the soil food web as the main character is how we are able to connect with the physical portion, the chemistry portion, better understand how all these players build the beautiful soil structure we want for our healthy plants in the soil. So, as well known, the Green Revolution, as much as advanced the way that we do agronomy and over the years, it also had a huge downfall that we lost a lot of biology in our soils. Again, not blaming anyone. We did the best we could with the science we had at the time, but now we cannot neglect that anymore. We are aware that those guys are important. They are the ones that really keep the system alive. So it's up to us understand them, harness them in our amendments, like Gerald is going to talk a bit more on part 2 of our webinar today. And how can we make those guys, or how, actually, let me correct that. How can we help those guys do all the magic for us under the soil? So just briefly, we here have a glance of how this full web works. We have all the nematodes. I'm going to try my best here. Let's see if I can get closer to Dr. Elaine. Bacteria, nematodes, fungi, protozoa, microarthropods. Not there yet. I need to train the speed, but you guys got the sense. And they are working in different layers, in different roles as they decompose the organic matter and the predators decompose the prey and the whole system grow over the trophic levels until we go up to above the ground, and then we have other areas studying that. Here at the Soil Food Web School, we focus on Dr. Elaine for decades of study, better understanding how a beneficial soil food web can help us implement real soil restoration. So, as you guys remember, many of you that already did our foundation courses, some of you are already completed the CLP courses and are on CTP or GTP program. Here at the school, we focus in 4 key groups of beneficial soil microorganisms. The first one of them that I want to talk about are the bacteria. We have insane diversity of bacteria in any soil around this planet. They are the first one to show up in any system and the last one to disappear when the system is heavily disturbed. They are one of the 2 main decomposers of organic matter, and they are capable of extracting the nutrients of this same organic matter from sand, silt, and clay. So they are our first explorers in any soil, and they help set up the stage for the other players to jump in when is the time. So, as prey, they are consumed by protozoa and nematodes, and they help build the soil structure by helping form the microaggregates. For those of you that are already advanced on FC courses or advanced on the CTP and GTP courses, when we talk about the liquid amendments, you heard a lot about letting the bacteria make the glue that will allow the biology to glue in the surface of the plant. That same glue the bacteria use to help build the microaggregates allied with the other players. And as more groups come into the place of your soil, you start to have the formation of macroaggregates and so on. With all this ballet working around in the soil, it's how those guys help retain the nutrients in a form the plants can absorb. So, as you guys know, and I will reinforce many times over the day, they are the ones converting any nutrient in a form plants cannot absorb to a format any plant can just take in and build up a healthy system. The second basic group are the fungi. Again, another insane diverse group. In each country or each, I think that microregion, we can go to the layer of microregion that you're gonna have opportunity to check the soil in your microscope. You will get endless possibility of diversity in fungi as well. Science is still starting to scratch our full understand around fungi, but we know that they are diverse in environments almost as much as bacteria. They also are responsible for decomposing the basic organic matter in the soil, and they also extract nutrients for organic matter, sand, silt, and clay. So as you can see, fungi and bacteria are the basic players that we need in any system to start reintroduction or restructuring the soil food web, in a healthy way. They are consumed by the nematodes, the fungal-feeding nematodes, and some microarthropods. So they also play a role as a prey in this system. They are very important to help the system structure the macroaggregates. So we have the bacteria organizing the microaggregates, and as the fungi enter the system with their hyper expansions, they will start to combine these microaggregates in macroaggregates, creating even bigger spaces and more structure in the soil, allowing a better water infiltration, a better niche to have all the other predators that we need in a health system, including macro arthropods, worms, and all the other beautiful creatures we need in a health system. And as I said, and they also retain nutrients in the soil in the form the plants can absorb. The other group that we cannot
Can I
forget that they,
interject a little bit
yes, please,
for a
go
second?
for it.
If
Yeah.
people want to know more about fungi, they are just quite a fascination to me. I don't know if anyone has read Stefano Mancuso's book, which is, I think, is The Incredible Journey of Plants. Have you read
Yeah.
it? It's what he's an Italian botanist, and he talks about plant neurobiology and plant intelligence, and how fungi plays a key role in that communication, how they're engineering underground communication, nutrient exchange. It's just really beautiful to see it from the other perspective as well. Sorry.
Yeah. No, please. It's on my wish list. I still have a bunch of books before that. But you
No
touched on a
problem.
very good point, Isadora. Thank you. Uh, the place that science are getting now with fungi, and here is my geek nerdy moment. It strikes me to that whole sense of everything is interconnected, like we have in the Avatar movie. You touch one side, you're going to touch the whole system in some point. So that's exactly what we expect to achieve in a health soil system. Everything is increasingly interconnected. And this is why when you work with one group, the cascade effect will come. So at the same time that this increases our responsibility, it also opens up space for us to speed up recovery for any system, right? Once you understand the logic and how those guys work for us, we can speed up the recovery of any system. Just need to take the time, understand, and on Dr. Elaine's word, just listen to Mother Nature. We have to stop fighting against it and work with nature. I love these words from her. They just like open up our eyes to so many factors that the system in place don't allow us to go for. So, on this sense, don't stop to be curious. Don't stop to reading, to look for information, to make good use of your community like we are building here at the school. You are not alone. And like Isadora beautifully said, alone we cannot do anything. So we also need to be a health web. Said that the protozoa, which on the aerobic side, the beneficial organisms as we classify them here at the school, we have the flagellates and amoeba, another insane diverse group. We can spend days in one sample in a microscope when you have a healthy sample. And I know that Isadora and Gerald are not going to let me lie alone. We spend hours and hours on microscope just chasing those guys, especially when you get them hunting or in the end stages of reproduction. It's just beautiful. We really hope that you guys indulge yourselves on time to time, giving you extra hours under the microscope, which also going to help increase the skills of doing assessments faster and faster and increasing the quality at the same time. So the protozoa, they will essentially consume bacteria in a higher volume, releasing the nutrients in plant available form. The famous poop loop that Dr. Lane brilliantly explained in the FC courses. And ciliates that are also protozoa, they also consume tons of bacteria, even if not more than the flagellates and amoeba due to their size, but they are indicators of lower oxygen conditions. So it's not that they're bad. They are also contributing to the nutrient cycling, but they are indicating us that our system is not on the best level of oxygen. What can we do? What should we do? Those are the kind of questions that we work during the CTP and GTP program. And they are also consumed by microarthropods and time to time by the omnivore nematodes because everyone is trying to survive. Down there is a huge jungle. So if they just like inadvertently pass nearby an omnivore nematode, they will be part of the feast. Which takes me to the nematodes as our fourth major group. We have four essential groups for nematodes that we classify them here at the school. We have the bacterial feeding nematodes, fungal feeding nematodes, the predatory nematodes, and the root feeding nematodes. Each one of them will also, each group of them will also contribute to the nutrient cycling, but they target different groups. The root feeding nematodes, we need to be careful when we are not when we look for them, sorry, when we identify them in our system, because this means that we need to understand where is the weakness of the system and how can we counter oppose to bring back the health and the balance to the system so they will not gonna have space or they're not going to have strength to attack the root system because we have predators chopping them down, and the root system is also more healthy, more robust to survive to a potential root-feeding nematodes attack. The predatory nematodes are on the micro level. They are the top notch of the predator chain. And like all of the other groups, they release the nutrients in the plant available form, helping retain the nutrients in the soil and contributing to a health food web, soil food web. So I think that I cover a lot of these topics, but let's do a summary. Bacteria, they decompose organic matter, and here in this video we have I think the original one is someone did making a protozoa infusion, but the system went to an anaerobic side. How do you know this is anaerobic? We have tons of bacilli bacteria, and I can say they're bacilli because they are long shaped, and these guys here, they are in chains. Do you see how they're like flapping wings in the system? So you know they have more than one unit. At this level, based only on this field of view, I would say this system is going towards an aerobic space. So I have to go back and check the entire scenario. What I'm talking about, where's the sample coming from, what's happening here before I can take any decision. And again, this is part of our journey here at the school, working with you guys so we can brainstorm and you learn how to counterattack cases like this of lower oxygen. The bacteria are consumed by protozoa, nematodes, and they help build the soil structure, forming the microaggregates, retaining the nutrients in the soil. So, fungi, I love them to the bones. It's just a surprise. You never know what you're going to find in one sample. And especially when you have beautiful long hyphae like this, you can just, I bet you, if you take a sample of the soil in the field and just a smell, I can guarantee that you're gonna have an incredible smell. You will be able to see with your open, naked eyes, sorry, the aggregates in the soil. So I bet to you that this is coming from a very healthy system, and I can guarantee that this is a fungi or the one of the top notch of beneficial fungi, because you can also see the famous clamp connections here. We have one here, another one being formed here, another one here, another one on this branch. So you have a beautiful coloration, diameters even all across the hyphae, septa evenly distributed. Those are the characteristics that we use to differentiate fungi from oomycetes. And here I'm going to take a chance to talk about oomycetes. Again, similar vibe as ciliates. They are not bad guys. They're just indicators of lower oxygen condition. Because we cannot forget that in the realm of fungi and the realm of oomycetes, we have potential disease causers in both of them. So just taking the chance to understand what's going on, you have to see how the plants are responding as well before taking by one image saying that's a disease causers. We need to be conscious that it's many layers of information that we need to evaluate before taking one final decision. Those guys are on the same level of bacteria on decomposing organic matter, extracting nutrients from organic matter, sand, silt, and clay, are basically consumed by fungal-feeding nematodes and microarthropods. So you don't want outbreak of microarthropods in your area as well, because they can chop down the fungal population more than you wish for. And beautifully building soil structure, forming the macro aggregates that we want so much. Oops, sorry. Okay. Here we can see one tested amoeba in action. Notice how the pseudopods are going out. Here we go. They are on a hunt. You can see how the body of the amoeba also moves inside, so you can see all the organelles, and pseudopod is just exploring the outside world. this one is active as they can be. Sometimes, or actually, I have to rephrase that because when we are doing assessment, we are shaking. And for us, this simple shake for them is a tsunami. So they tend to just slow down their activity. So, time to time, prep a slide and let the slide sit for half an hour. For fun, just go and check because you might be able to see those guys in action. They consume tons of bacteria over their lifetime, releasing the nutrients in the plant available form via the poop loop. And again, if you have ciliates, they are indicator that your system is going towards the lower oxygen condition. Just don't take this decision based in one field of view only. Always check the entire assessment, the entire readings, all the readings before making this conclusion. And they are consumed by microarthropods. So it's really a jungle happening there. And you might even be lucky to find microarthropods preying on those amoeba. And if you find, please share with us. Contact your mentor. If you don't have a mentor, please email info@soilfoodweb.com, sharing your videos and images. We love them, and your images and videos can be posted in our microbe library as well, helping other students see the beauty images you are having in your area. Nematodes, another cute ones. I didn't say cute much yet today, did I, Isadora? I don't think so. So
Yeah, not yet, don't worry. They're cute too.
my students all the time I joke with them like how many cute I said today. So um, you will learn with us how to differentiate the four main groups based on the mouth parts of the nematodes. Here at the school, we don't worry much about species, even though when you are working with a crop and you have known organisms that are disease causers, of course, you're going to look information about them. But to identify how healthy your food web, your soil food web is, we need to understand the ecological role. And in nematodes' case, understanding their ecological role is to understand what they're feeding on. So for this, we classify them as bacterial feeding nematodes, fungal feeding nematodes, predatory and root feeding nematodes. Like I said, all of them will be cycling nutrients. The predator nematodes are the top notch of the predator layer. And the root feeding nematodes are the ones we don't want because when I'm growing something, I want to have a yield. And if I start to have a lot of root feeding nematodes in my system, I will lose productivity. But if I work to have a healthy soil food web, those guys will not gonna have space to expand their population and reach a point that they can cause too much damage to my system. Oh, here we have an omnivore nematode. Look how beautiful they are. And again, huge diversity in shapes, sizes, and forms. Of course, that the omnivore and predatory nematodes tend to be bigger than bacterial and fungal feeding nematodes. And how do we know that's omnivore nematodes? Checking their mouth parts. Here, even though looks like we have a chamber mouth, notice how the chamber is not so deep. It's wide, but it's shallow. So this, imagine a space that, do they have enough area to hold another nematode inside of that mouth until they can suck in all the interns? Probably not. So high chances this will be an omnivore, and you have to cross-check all the other characteristics to decide which group or which ecological role this nematode is working on. And as you can see, this is how we work the decision on how to identify the microorganisms here at the school. Which ecological role? Yep.
Sorry. Yeah, do you know there's 80% of all animals on planet are nematodes. There is just, it's amazing. It says,
Yes.
I don't know about this data, but it says 60 billion nematodes per human on Earth. So there is a lot of them. There is people who study a whole career. I have a Facebook group that they tell me exactly who it is because I don't know it as much. But yes,
Yeah.
they are super diverse.
Yeah, great point, Isadora. And this huge majority of them are inside our bodies. So if we have healthy soil food web, we're gonna bring the good guys to inside our body and help replenish that. And another fun fact, Dr. Elaine husband is a nematologist, and he worked his entire career understanding how those guys help healthy soil food web. So a lot of his knowledge is also applied here at the school. And like Isadora said, it's just like we have nerds in every field, thankfully. So look for those groups nearby. They are also part of our community. So let's build together. When you learn, share the groups you learn about on our forums. We have the FC forum, we have the CTP forum. That's also part of building your community and help us expand this with other students as well. Great points, Isadora. Thank you. Fungal feeding nematodes. A lot of people get scared about them that, hey, Carla, they can puncture the root system as well. No, because they only have the stylet, as you can see here. Imagine yourself trying to punch like just a thin wall. You don't need much strength, right? Just one small punch and you are done. This is what we have in the fungal wall when you compare the size of a nematode with a fungal hyphae. They don't need extra strength. So just the pressure that they have to launch the spear outside is enough to puncture a fungal wall. So they will just puncture the wall and suck it up all the hyphae interns as they had the straw in their mouth. And that's how they're gonna have their milkshake in the end of the day. You're only gonna have fungal feeding nematodes in your system if you have a minimal viable amount of fungi. So as you can understand, as you can see, is as if you grow your fungal population, you're going to start to speed up or advance on the presence of nematodes. Because like any food web, if you don't have a minimal biomass of prey, you cannot sustain the predators. So this is very important for us to understand when deciding how much of each amendment we need to apply to recover each group is necessary in the system. Oh, I love this video to the bones. We have that in our CLP courses. I hope you can see here the naked amoeba, how here we go again, how they're playing the environment and scavenging to where they're going, exploring the path. And you can see all the organelles moving around. And this is a huge naked amoeba. I just wanted to give you another extra tip is that until you see naked amoeba the first time on your microscope, you're always going to be guessing if you saw one. When you see one, even if you had to start the assessment again, just go after this guy. Chase them to see how they move, how they feed, how they work, all the environment around them. It's worth start the assessment again after that. Oh, here we go. Rotifers. A lot of people talk about rotifers and the beautiful what bears or tardigrades, the incredible almighty guys that went to space, came back to Earth, and they were alive. Sammy has a huge crush on them. It's also her icon on social media. I like them a lot as well. They are a cute kind called water bear because when you see them walking in your slide, they look like cute little bears everywhere, like crawling your sample. And here we have a rotifer. Rotifers are not protozoa. They are multicellular organisms that at the same time they eat a lot of bacteria. They thrive better in lower oxygen conditions. So we don't want many of them in our sample, but if they're there, we just gonna put our red alert on checking what's happening in my system that they are able to thrive. So they're not bad guys. They are just our environmental indicators. Okay, stock ciliate is a ciliate, and this video is incredible because you can see all the swirl happening. Can you see the bacteria moving in circles here and this side? So they just have like two small helicopters in their head, spinning around. And as this water spins around them, they're going to filter in the bacteria inside their body, leaving the water outside, and they will digest the bacteria in the organelles. Again, lower oxygen condition indicators, but cycle nutrients like crazy. The stocked ciliates, they will be connected in the microaggregates until they decide that it's time to move on. Then they will just detach from the microaggregate, let the water movement take them to another direction, glue on the surface again, and start the entire process. So as you can see, diversity in soil food web is just gorgeous. And here we have a nematode egg almost to the point to hatch. As you can see, the nematode is moving inside the egg shell. And it's similar to a snake egg. So if you saw a snake egg before in your life, it's that same flexible structure with the nematode being formed inside. When this guy is ready to hatch, it's just gonna through the enzymatic process, you're just gonna open up the space over the shell and get to the outside of the environment looking for food.
Carla,
So
I saw
yeah,
one a nematode birthing a nematode egg. It was pretty interesting and cool.
awesome, beautiful. Probably you can see Isadora images and videos in the microbe library, so spend some time there. And said that, Gerald, I pass to you the honor to guide you through the gathering beneficial microorganisms in each one's area.
Thank you very much, Carla, for giving us such a great introduction to the soil food web microorganisms. We always enjoy looking at all those little critters and all the different things that they do. Um, After that great explanation from Carla, I'm gonna talk you guys through how we cultivate and how we gather those microorganisms in our system. So first of all, let me tell you some really interesting facts. First of all, I want to remind you that we are never alone. In fact, we are actually kind of like an ecosystem. And let me explain that. If we take a look at our human bodies, it has been discovered that it's actually composed of both human and microbial cells. In your body, you carry more microbial cells than human cells, roughly 39 trillion microbial cells against 30 trillion human cells. So actually, you are more a microbe than a human. And actually, there is a great book about that we were talking about a couple of few weeks ago when we were in the workshop in London. The book is called 10% Human, and it talks about how we are basically a colony of microorganisms and how those microorganisms influence many things happening in our bodies. So again, technically, we are formed by microbes. We are a walking colony, and those microorganisms influence our immune system, even our mood. That is fascinating, and I think that's one of the biggest reasons why we should be paying attention to these guys, because it's not only about growing crops, it's also about cultivating our health in general, our food security, growing stronger since we are kids, right? So that's a very fascinating, number to share with you. Also, if you take a look at your skin, you can have about 1,000 species of bacteria just on the surface of your skin. That's another crazy fact. Think about what 1,000 people is. Think about all those species in the bacteria, in your skin, sorry, coming from the different microbial life. That's fascinating. Also, the air we breathe is alive as well. It's alive. It's full of bacteria, full of microbes. Um, every day, humans inhale about 1,000 or 10 billion mold spores. But all these things should not, be scary for us. We've commonly heard you need to wash your hands, you need to be careful of this and that. Microorganisms are sometimes approached from that perspective that they can only be harmful And whereas it is true that some of them can be harmful for our health, the vast majority of them is actually helping us with our daily activities. And it's important to take care of that microbiome that we have in our body. One of the best ways of taking care of that is cultivating foods or generating spaces and soils that contain those microorganisms as well and that are healthy. So basically, when we talk about regenerating soil or making compost, we're basically just mimicking what nature does in our body, in our forests, in our lakes, everywhere, because microorganisms are the drivers of all the different systems. They are the ones who carry on with the different cycles that keep things alive. Think about an old forest. An old forest has been around for thousands of years without any human intervention, luckily, and they never have to be fertilized. They never have to be sprayed with any chemicals. What's the reason for that? Because the microorganisms are constantly, renewing the energies, giving the plants and the insects and the animals all things they need to carry on and continue their cycles. So I mentioned this because it's fascinating. All what we are doing with our approach is trying to enhance those microbial systems in our planet. Um, so let's talk about where we find them in nature. I just wanted to confirm if everyone can hear me well, or if you guys are struggling to hear what I'm saying. Okay,
I hear
great.
you fine. Yep.
Excellent. By the way, I saw a great comment in the chat. It says, I eat microbes on my food every day. That's true. And that's the goal. We're not always eating microbes because sometimes the quality of the food that we get might not be the best. But that should be the goal. Eating microbes every day so that you nourish your body and so that you become a healthier person.
We have a comment here that your voice is very soothing, just like Morgan Freeman of Microbes.
The Morgan Freeman of microbes. Okay, that sounds cool. Yeah. All right.
Okay,
So
remember to bring that to mentors meetings, Adora.
Okay.
there you go. Another nickname for me. It's been like the fifth one this month. Okay. Thanks a lot for that. That's very sweet of whoever wrote that. Yeah. So in nature, the idea is to to find those places in our system that contain those microorganisms so that we can start cultivating them and, increasing the health of our soil, our plants, and increasing our health and our productivity in general. So in nature, we can find these microbes on animal skin as well, just as our skin. In manures, yeah, in animal manures. For most people, manures are disgusting. I'm actually one of those weird people who like smelling manures to get a feeling about how good they are. And if I am composting manures, I love the smell of their decomposition. I know that sounds weird, but it's because I know they are full of life and I really enjoy, being in that kind of environment. So even though manures have been considered disgusting, just waste. Some people don't even know what to do with them. Okay, here we go. The Morgan Freeman of manure. All right, yeah, sounds good. Okay, so that's the sixth nickname this month. Let's see if we can get up to 10, so you guys can come up with your nicknames there. I'm happy to hear those. uh, yeah, so manures. We can actually convert manures into something great for our system. We can use that for nourishing our soil, which in turn will nourish our plants and our bodies. So we can also find the organisms on the surfaces of leaves, stems, branches, bark, and roots. Again, think about those forest ecosystems where we have all those organic matter depositions. Everything that we find in our system that mimics those organic matter, sources are going to be food and house for microorganisms. And of course, in the soil, but not in any soil, only in healthy soils. So if you know that your soil is not healthy, you should be looking for ways of increasing the microbial life and really, enhancing the microbial properties, because that way you can really recover your system and get more benefits. Um, so where do we find these guys for soil regeneration? Basically, if you're a human, you need basic things. You need food, water, you need a nice temperature. Well, Isadora might be struggling with temperatures now, but you do need a nice temperature. Um, so we can find them in those specific places. Where do we find those conditions? Okay, they like living in decaying plant matter, whether it is dead leaves, grasses, woody materials, everything that was once alive will be consumed by the organisms, even ourselves. When we die, we're going to be decomposed like any plant or any tree that dies. The microorganisms are going to take care of that. So these are the places that we can collect the microorganisms from. The idea is that we gather the good sources of materials and we put them together into a composting recipe. Then we manage that compost properly so that we develop aerobic conditions, meaning that they are full of oxygen. And there is a whole process where the compost is going to heat up and then it starts to mature. And it's fascinating because we can track the changes in the microbial populations in that compost as it ages. And then we will find that at some point during the maturation, we have the full set of microbiology that Carla was talking about. So something that I wanted to emphasize on here is this slide, for example, says compost aerobically. We often hear the word compost, and it's a very common one. But here we're not talking about any type of compost. We are talking about compost as a source of inoculum. That's our main focus here. Actually, many of us say the organic matter or the compost itself is just a carrier. It's just a secondary benefit that we're obtaining. But the main benefit that we want to cultivate in our compost is the biology. And let me tell you, it's not any recipe, it's not any set of materials, and it's not any process that gives you high quality compost, high quality inoculum. You really have to be careful about the way you are making it, about the proportions of the different materials, so that you can really get to a high quality product, because we know it's not just organic matter. It's basically what lives in that organic matter, what we need. So very important. These are the sources, but we need to make sure that we take care of those guys. Actually, we see compost as a living process, not just processing waste to make it turn into a nice color and be able to use it for something else. No, it's not that. It's cultivating the microorganisms and facilitating the conditions for them to thrive. Yes, Isa?
Someone asked in the chat if does compost, when done in soil food web way, ever become anaerobic? And this is an interesting question in a sense because remember the microbes that Carla, sorry, was mentioning before. We're talking about a very holistic approach, a very complete soil food web, especially fungi. Fungi is what really needed in soil, and those guys are aerobic. And this is why we thrive and we work really hard to get the right materials, to control those situations, to make sure our compost is aerobic. It doesn't mean that the other one is bad, but for our approach, for the way we are aiming, we definitely need this to be aerobic. We need to control the temperatures, the moisture, and the air. But this next week, we'll talk more about it. But just thought it was a good question.
Stay tuned. Yeah, that's a great answer, Isa. Again, our purpose here is to build soil, and for building soil, we're mainly building the aerobic populations of microorganisms. That's why we emphasize a lot on having the right proportion of things, the right porosity in our piles, so that we really develop the conditions for those beneficial guys to thrive. Very important, if you want to gather these materials from your own region, that is the best thing to do, okay? Think about myself. Again, I am in the beautiful Costa Rica, tropical conditions. Think about our country in March. Nice temperatures, warm, long days. If you take me all of a sudden to Norway or Denmark or Sweden, in March, I see some of you coming from Sweden and Denmark. If you guys take me to your countries in March, I'm gonna struggle because I don't have the clothes, because I'm not used to those temperatures, because I'm used to different foods even. So I'm gonna struggle. It's the same thing with the microorganisms. If you collect them from your local area, they are already used to the tropical Costa Rican weather, or to the Denmark weather, or South Africa, or Ecuador, Australia. Whenever you guys are joining from, you have your sets of microorganisms already there, and they are used to the foods, to the conditions that you guys have over there. So it's always better to get them from your region. That's going to make the process easier. All right. Very interesting. When we go to different sources about composting, you will find this concept a lot. The carbon to nitrogen ratio is basically the proportion of carbon to nitrogen that each of these materials has. And in many situations, you will find that you need like an average or an overall 0 ratio to start the composting process without even making a distinction in different categories. In the Soil Food Web, we do it slightly differently. We are aware of the C2N ratios, but we use them to classify the materials into 3 different categories, knowing what each of the categories will give us. One of the categories is high nitrogen materials, which have a ratio that goes from 5 to 25 to 1. That means they have a lot of nitrogen, and that means one thing. Then we have the green materials, which are like the most balanced. We could say that many of them have a 0 ratio from 25 to 1 all the way up to 60 to 1. We like to say that a good green material is going to average a 0 ratio of 30 to 1. And then we have the brown and woody components of our compost piles, which are very high in carbon, and their 0 ratio goes over 60 to 1. Why do we make this distinction? It's basically because we are, again, building compost piles for inoculum. We know that we need the bacteria, the fungi, the protozoa that Carla was talking about, and by using different sets of materials, we are stimulating different populations. Just like humans, again, different organisms prefer different foods. So if you have a good diversity of these materials and you have a good proportion of them in your piles, some of them will be preferred by the fungi, some others will be preferred by the bacteria. If you have the bacteria, you will stimulate certain types of nematodes and protozoa. If you have the fungi, you will stimulate some microarthropods and some fungal feeding nematodes. So basically your foods are influencing the different things that you're bringing to your compost pile, to your inoculum source, right? Also, it's not only having a diversity of these 3 categories, it's also having diversity within each of those categories. Why? If you have different types of greens, there's going to be different types of bacteria associated to those different green materials. The same if you have different types of woody materials, they are going to prefer different types of foods, and that way you bring the diversity. Why is diversity important? The more diverse a system is, the more resilient it will behave, okay? Again, because some bacteria will be used to a certain set of moisture in the soil or a certain type of temperature range. So all that is going to give you different benefits, and it's going to make your system more resilient. Firstly, in the compost, secondly, in your soil. So very important to consider that diversity of materials. Here we have like a rough explanation of the different types of materials. In our courses, you get more in-depth understanding of all this. We will talk about each of the types in more detail. For example, high nitrogen. In high nitrogen materials, we have fresh manure, beer mash, which is very common in some places in the US, for example. Seed germs, which can be really, really powerful for creating heat in a pile. Legume plants, depending on how well they are growing, and also the time of the year. For example, alfalfa, different types of beans, chickpea, any legume plant that you can think of, it can be potentially a high nitrogen. Then we have examples of green materials. These have, for example, the grasses, the green leaves, veggie scraps, which are really fun to work with, non-legume plants, used coffee grounds. And then we have the high carbon materials such as wood chips, straw, coconut husk, dry leaves, paper, cardboard. And as you can see, the bacteria tend to prefer the high nitrogen and the green, whereas the fungi prefer the woody or brown components of that compost recipe. So again, it's about this balance that I was mentioning. These different foods will stimulate different groups of decomposers, and then those decomposers will start stimulating their predators, and that way we start, enhancing our system. Okay. There's a couple of materials that we also want to avoid or limit, since we are cultivating soil health and our health in general. We have to be careful of which materials we use. For example, if we know that the materials have been sprayed with pesticides, insecticides, herbicides, or have been produced using excessive inorganic fertilizers, we know that all those things can affect the biology. And again, since our focus is inoculum, we're not going to get any inoculum from that. We can actually be introducing substances that are going to kill the biology that we have in our compost. So we have to be very careful about how our materials were managed. We also want to avoid materials that inhibit the growth of microbial populations. For example, things coming from pine, eucalyptus. Another example is pampa grass. If you know that you are dealing with those materials, it doesn't mean that you cannot compost them, but maybe they need a certain management. And the best idea is not going to be to use them when fresh, because they can be harmful for biology. So these materials, we have to be careful with the management that we give to them before they go into a compost pile. We also want to avoid manures coming from animals that were dewormed or fed with pellets or high salts, because again, all that is going to affect the microbiology. Also, we want to limit soil or organic matter from local areas. Why is that? Well, basically because we want our system to be environmentally sustainable. So we don't want to over-collect or cause any damage or imbalance in our natural systems. You can certainly pick a couple of handfuls or a couple of, leaves or bark or whatever you have, close to start stimulating your inoculum, but you don't want to overdo that. Otherwise, you will alter your natural ecosystems.
Thank you. Thank you so much, Gerald. Thank you so much, Carla, for explaining this very complicated thing, because the truth is we really don't know that much about them, right? But we just know enough. We don't have to micromanage microbes, but we just know enough that we need a lot of them. We need a lot of diversity. And we want to create the right environment for them, because like Gerald very wisely says, they need food, they need a house, they need the right conditions. No one likes to be in the heat. I don't like it. They don't like it either. If we don't put plants on soil, they're going to have a lot of heat. So we're just gonna show, while we get our heads around all this, our promotion for this month. Do I click on it? I don't know.
Some of the farmers working with Soil Food Web consultants around the world are seeing remarkable results.
Just within the first 3 years of working with these folks, they saved over $1 million just by cutting back on synthetic fertilizers, going from 200 units of nitrogen per acre down to 150 to even 50 units of nitrogen with the same, if not a better yield, which is really exciting.
if we look at the fertilizer savings across 1 hectare, we're looking at about $1,000 in savings. And then when we look at the increased revenue from the yield increases, we're looking at about $504. So the gross margin was increased by about $1,500. And then if you extrapolate this over 200 hectares, we're looking at about $300,000 over the course of any given year.
You can see the average again, 72%. And if you just take a few of those examples, you can take celery or you can take kale, you can see that we reach over 100% of yield increase.
Basically, the weight gain again was like 25% or more. The only complaint they got was the client wasn't used to obviously bailing as often because the crop was growing so much better and so much healthier. They went from 280 to well over 400 sacks an acre. So that's 42% increase, which is really impressive. I mean, dollars and cents wise, that's a big jump. The PEs were really amazing. They were really aggressive with some of the PE projects. 136% increase, that's pretty impressive.
With the Regenerative Farmer Package, you can register for the Soil Food Web Foundation courses with Dr. Elaine Ingham for just $31.99, saving over $1,800. Plus Stage 1 of the Grower Training Program worth $2,000 for free. Register before midnight July 17th to take advantage of the early bird discount and save an additional $200. There are only 50 places available in the summer 2025 registration cycle, so please don't delay. At the Soil Food Web School, we are so confident in the quality of our courses, we proudly offer a 100% risk-free guarantee. Here's how it works. From the date of your registration, you'll enjoy a 30-day window with a no-questions-asked money-back guarantee on the total price you've paid for our courses. The only condition is that you cannot have watched more than 32 lectures of the foundation courses. Your satisfaction is our priority, and we stand behind our commitment to your success. Finance options are now available, so you can pay at your own pace with a firm. We look forward to welcoming you to the Soil Food Web community.
All right. So I hope everyone is well. Everyone is doing okay, and we're going to start with some of the questions that... No, thank you. We're going to start with some of the questions that you guys have been asking us. I can read out the questions, and Carla, Gerald, feel free to answer. We can all answer. We can all have a go with it. It's all good. So first question comes from Kelly, and she says, where I live, the soil is very clay-like and dry. Oh yes, my yard cracks. I know those soils. Please address how to help this so I can grow veggies and other plants without having them to die quickly. I would love to know how to compost without getting ants and flies all over the yard. That's a
start to that?
Right?
Yeah. Do you want to start, Isa, since you feel the pain so close?
I do. I feel the pain. I see clay soils a lot here, and yeah, it's not balanced. And I mean, it's unrealistic to say let's get a big truckload of sand and balance that, geology out of here. But I have observed that under the right conditions, with the right amount of organic matter and biology, it definitely acts. It doesn't crack. It doesn't go like this. So it is a matter of just building organic matter and getting more biology back in soil. It's a very generic answer. I'm so sorry. I don't know your context, but I don't know if you guys have anything better to add to this.
Yeah, normally I encourage folks, and it's what I do in my work, my jobs, is that you don't need to use only biologically active compost to bring up organic matter to the soil. In fact, we have so many different types of composting that in the end of the day, if they're done properly, meaning you know what's coming in and what's going out, you can use this less expensive compost to increase the amount of organic matter in the soil. And then you complement or you bring the biology with the thermophilic process that we use here at the school. So it's just a matter of figure out first the budget, how deep the problem is. And it's not a one-time fixed solution. Like Isadora said, you have to have multiple inputs and multiple growing seasons actually to really bring to the best place you can get. What did we miss, Gerald?
Yeah, another thing that I would add is covering the soil, making sure that the soil is covered, throughout the year if possible, by using mulch or cover crops because of several things. First, if you have, for example, plants, they're going to work as solar panels that will capture the carbon from the atmosphere and will have the potential of increasing your organic matter. And they can also sustain life. So they will enhance the microbial populations and they will help the system be alive. And actually, it can even help the system move faster in those types of, heavy soils. That's something that I would add to what you already said. So basically, ways of increasing organic matter, inoculating that soil with beneficial biology, and keeping the soil covered. Also remember, if you have the biology and the organic matter, you are going to improve the water holding capacity of your soil. So eventually, you will be getting different benefits from this type of management. What you say about compost, okay, we need to understand that certain materials will attract flies, and it's almost impossible to get rid of them sometimes. An example is chicken manure or veggie scraps. They tend to love
Beer
those.
mash.
Yeah, beer mash. However, what I've observed with the thermophilic process that we do is they will be present in the first, let's say, week or two, but then as the pile continues evolving and maturing, they will eventually disappear. The issue might be if you have a system that is static or if your recipe is imbalanced, because that's going to constantly create the conditions for those flies to come. With thermophilic composting, they might be present at some point, then they lose their interest in their pile. But if you're using a static compost system or if your recipe has excessive green or high nitrogen materials, it can create the conditions for the flies to continue coming. That's what I would add to that answer that you already did a great job with.
Yeah, for sure. Yeah, just remember this. I mean, clay is the smallest particle, is like bacteria, is really small. When you're talking about dry, it just gets really compacted. There's no oxygen there. So if you don't have oxygen, that's your limiting factor there. So think about perhaps when you're starting to plant, because leaving roots is definitely the best way to go. Try to aerate this somehow, or just find the right time and place to plant it. But for sure, do you guys notice that the flies are attracted to when I add dry bone blood meal?
meal?
Yeah, it
Yep.
really attracts the flies. I don't know if they really like the nitrogen kind of thing. It's quite interesting.
Yeah. And beer mash for me is just like the champion on flies attraction, but I love the results. Nematodes just go wild. And another player that you can bring to the system are the worms. They will help build up the soil structure. At the same time, they plenish the biology as well.
True, true. Okay, so Suze, I really hope I'm not butchering anyone's name. I know my name is hard and people say my name in very different ways, but it's with love, please. So do different crops. I'm working with fruit trees, need the same compost biology soil.
Simple answer, yes. They don't need, sorry, the answer is no. Come on. They don't need the same compost biology in the soil. It's really understand, like Gerald said, you don't want to grab someone from Costa Rica on summer and fly them to Denmark in the middle of winter without no jackets. So why bother to all of the risk that you're harnessing all the biology in your good compost and apply in a different microregion or microbiome, risking to kill all those good guys. So the best procedure is what Jared brilliantly explained to us. Use local material to build your local compost. That's the one that are gonna get you faster results. It's not that using from different regions you're not gonna bring a good. It's just gonna take longer. And each type of plant, meaning slash crop, they will be in a different succession stage of ecological evolution. So you need to understand where your crop lies on. So you're going to be able to figure out the best level of each of the four main groups of microbiology you need in your soil. We work a lot with the fungal to bacterial ratio, because if we have those guys balanced for the desired crop, the other players will come in place as the interaction between the root system releasing all the exudates, or will attract or will boost up a specific group of biology in this synergy that's happening constantly in the soil. So as long as you let the system work for you and pay attention on details to implement or to boost up what is missing, the balance will get in place and you reach a point of just maintenance. So it's time and observation and patience. What did I miss, Gerald and Isa?
I think that was a very complete answer. Yeah, you were talking about the FB ratio that's related to succession. Different plants or different crops that we grow belong to different stages of succession. If you have trees, then that's a more advanced stage of succession. And those species prefer fungal dominated soils for different reasons. One of them being the type of nitrogen that they prefer, and also the nutrient pulses that they need. The nutrient pulses are different in an early successional system than for example, in a fruit tree system where the species are sorry, it's not annual, it's the other concept. What's the name of the other one?
Oh,
Perennial,
gosh.
yeah,
Yes,
perennial.
thank you.
So they need different types of nutrient pulses. So yeah, it's about FB ratio. Sometimes you cannot make the compost that necessarily matches what you need in your soil, but at least you know that you need to favor the growth of certain decomposers.
Yeah, I think at the end of the day is understanding that they need to be there, and our management style for the past years haven't been taking them into consideration. So usually you're dealing with systems that have nothing. And the easiest way to see this is what is growing naturally in your soil, right? There are wonderful books when Weeds Talk or Gerard Ducerf, he's French, is more here for European people. They talk about bioindicators, or all those plants that are naturally growing in a system, and it's telling you because nature is always trying to, to get a balance of the situation, and then so those are interesting things that let us know what's the baseline, and that's going to be important to understand what's happening over there, right? What is the baseline for everything? But yes, and everything else.
Yeah, we have a very interesting comment by Dr. Tony in the chat that the pH of the compost produces important to determine the sustainability of certain crops. And that's a good relationship between berries and low pH and substrate. Exactly that. So it's part to what we're saying so far, like understand the recipe so you can balance the compost to reach the desired point for your related crop so you can cut to the chase and have the best compost available for you. Good point, Dr. Tony. Thank you.
Oh, sorry, someone already changed it.
No, it was not me.
Because I was doing two things at the same time. I don't.
Yeah.
Okay, so Herman, he says, how negatively does applying artificial fertilizers affect the soil biology in soil? Can small amounts still be being used without too much negative effects? That's a lovely question. Love it.
That's a controversial question. Can I start?
Go ahead.
I already did. Sorry, guys. I should have asked first. So, the artificial fertilizers, it really depends on which one you're talking about and the amount you're applying. The main issue with those fertilizers is that when they reach a point of application, they start to behave as salts. And when behaving as salt, they will take out the water of the system, and this will directly affect the survival of the biology. And it accumulates in time. And without biology, the system will gonna fall crack through keeping the structure, keeping the water infiltration in place. So it's a cascade effect that we notice over the years. Gerald can talk more about this as well, but when we are working on a transition system coming from the traditional place where we use the artificial fertilizers to a more regenerative ag approach, you also, in some cases, you might still need to use some of those fertilizers, but it's good to understand how they will affect your local biology. So it's a lot of study and testing in small areas so you can see what you can do. But most important is on this transition period, look for alternatives. Do you have natural fertilizers that you can use allied with the biological compost that will be less damageful than the artificial fertilizer? Those are the kind of alternatives that we are looking for when talking about regenerative ag. It's a brainstorm and endless learning curve. What else, Gerald?
Sorry, I was muted. I agree
No.
completely, Carla. I think you summarized it in a very good way. We have to keep in mind that our ultimate goal is to only use biology and only use biological amendments. That's our goal. But sometimes getting to that goal is not as easy because sometimes we start from a very depleted system, from a very depleted soil that is lacking, for example, some minerals, or it might be lacking organic matter or biology. And those three things have to work together for your system. You cannot continue the transition if any of them is not present. And sometimes that will imply that you need to use some level of the fertilizer that you were applying, or as you said, you can find alternatives. Uh, there is different options for remineralizing a soil, which is a concept that I like talking about. So yeah, it's kind of okay, at least decreasing the inputs or getting rid of the most harmful ones at the beginning and trying to replace that with other alternatives as you build your soil biology. Mhm.
Awesome. Yeah, I have nothing else. I don't feel like we need all to chip in because this was brilliant. I agree totally with both of you. We can shift biological communities in soil, but at the same time, we have to understand what is that starting point because agriculture is, you need a livelihood from this, right? And we need transition. So there are transition periods that we should not be so, closed-minded that we're not going to do this, we're not going to do that. We just need to find the right path and we need to have an end goal that is very clear. And yeah, so everything can be valid as long as we understand the consequences. Are we taking 2 steps back? Can we go 3 steps forward? I don't know. It's all very contextual.
Very good point.
Anyway, Tim has asked, have you done any work with fungi to remove contaminated soils? I have read a few articles. Interesting. I haven't particularly worked with that. But I feel like they're always decontaminating something in somehow. I feel there's always microplastics and everything else. So as long as we get enough fungi in soil, we're just going to benefit from it. I don't know if you guys had any particular job or work that you did that was specifically for remediation, bioremediation.
Hey, they are one of the main organisms that we use in soil remediation. They are easy to multiply, easy to apply, easy to see the answers they are sending us to the system if we need to add more, if not. They are relatively fast and makes the process cheaper because it's easy to multiply. You just need, you don't need to be like sterile lab for any of those things. So, but like I said earlier, the science is just scratching out the surface of the endless potential things we can achieve with fungi. There is many people working with that. One person that is doing a beautiful job on presenting all the power of the fungi realm is Paul Stamets, and I highly encourage you to look for his publications to better understand the precise mechanism for specific species or for specific groups when working with contaminated soil. Especially because this kind of job is crazy expensive, demands a huge team of multidisciplinary folks. So more information we can advance using biological approach, easier will be. And fungi will just, when they are not needed in the system anymore, they will self-control their population size. And I don't see any backslash unless is a potential exotic fungal species that we don't know how they will respond once the decontamination process is done. So, like anything related with biology work, you need to fully understand why you are using this, why that specific species, and which are the potential damages if they have outbreak in the system, meaning they don't have a natural predator or natural control keeping them in check. So just be careful with using the isolated microorganisms in general, because instead of fixing one part, you might be bringing bigger damage for you in the near future. So just read carefully before deciding apply anything. Gerald?
Great answers by Carla and Isa. I just wanted to add, Tim, that this is a really fascinating topic. Like 5 or 4 months ago, we were giving away some inoculum from the oyster mushroom, and it's known for being edible. And we were like, well, what are we going to do with this in a compost? But then we started reading scientific articles about it, and it has the potential of accumulating heavy metals, or decreasing the heavy metal contents in the soil. It also has the potential of breaking down pesticides. So what we are now doing is we included some of that inoculum in a compost pile to first test how it can, adapt to a compost environment in terms of survival and competition with other types of fungi. And then we will see if we can test. We can test small first and see if it can reduce the contaminants in the soil. It takes time. It's a process that takes time because you first need to get that compost, and it takes a while. This recipe that we are trying is fungal food dominated, so it takes a while because fungal foods are usually slower to get decomposed. So yeah, we are still in the process, but if we discover anything important at some point, we'll keep you updated. Fascinating topic, yeah.
Just remember that the system will select as well. Though this is what Gerald is saying, it takes time, right? It's not like, oh, I'm going to put those in there. It happens a lot with mycorrhizae, in the beneficial. There's a lot of spores out there. We buy it. But if your system is really primary, it's maybe not ready for those poor mycorrhizae. So you might spend a lot of money and you still need to work, a little bit more. So it's a process and there's stages, and so think about that. All right, Jenny. In urban situations, growing areas are surrounded by concrete and asphalt. Will protozoa and nematodes show up? Yes, they will.
Trust the system. They
Yeah,
will. They
they
are
will.
ferocious, and the concrete and asphalt, it's normally just up to 20 centimeters top. And below that, you tend to have 20, 40, 60 centimeters, depending on the design of the area, if it's a road, patio, whatever, to sustain this paved layer. But and it's a heavy mechanical compaction. So those layers that really build to sustain the patios or the roads or whatever similar to that, below that layer, if you have any interaction with root system, those guys will thrive. If not, they will be present, but not large volume because there is no much reason for them to be there. There is no food for them. Like any living organism, they are looking for food. If they are starving to death, or they're gonna go dormant in cyst format, or gonna look for another place to survive. Simple survival skill.
And there's birds flying as well, bees, the amount of organisms trapped in a little bee, and in the wind and in raindrops. So yes, there is a power to inoculate everything, even though it doesn't look like it, but they do appear.
Yep.
It's really magical.
The power of invisible.
Anything else to add, Gerald?
No, I'm good, thanks.
Right. Don't want to leave being left out. All right, next question is, what is the best way to prepare land to make sure that when the biology is applied, plants can thrive? For instance, I moved to a new house with poor soils. I applied high-quality extracts to vegetable seedlings, which has kept them alive, but they weren't thriving because the soil is so poor. Crude reality.
Do you want to summarize the
Yeah.
course in 2 minutes, Gerald?
Well, I think it's mainly about time. Isa was talking about, I think about the environment. The current environment doesn't necessarily have the conditions for all biology to establish. So maybe in the first application, some of those organisms will survive, some of them will establish, but it's not going to be enough. So the plant is still not responding, maybe. You do another application, so some more get established, and so on and so forth. So it takes time. 2, 3 applications might not be enough. It might have to be 6, for example, and those 6 have to be accompanied by other practices that support the biology. So as we were saying, making sure that you have good organic matter, that you don't disturb your soil excessively, making sure that you have the right moisture, all that is going to play a role. But the idea is this can take time, depending on how disturbed the system usually is and how intense the application program is. So maybe it's a matter of time. Again, we also have to think about the mineral part of the soil, the organic matter in the soil that plays a role. But from a biological perspective, I think that might be an explanation for that.
Yeah, and I think it's important other topics that I talked about earlier, like have the cover crops interacting with the vegetables, have a diversity of vegetables in the first years in the area, because you're going to have different depths of root system working around. So biology, we're going to go over more depth of the soil. So those kind of little tricks that Gerald explained to us earlier as well, that can help you boost up the system. But patience is the key, like setting up expectations. It's a multiple growing seasons to see system that's coming from a very poor condition, or like Dr. Elaine says, coming from dirt to return to soil conditions. But don't give up. Those guys will show up. They will work for you with happiness. Just give them time and give them some root system to work with.
Yeah, I mean, just think that they're not there for a reason. So environment might be hostile still, right? Like think about the most famous athlete that you can think of. No matter how great that person is, we can send him to the most inhospitable place that we can think of, with no shelter, no food source, no family members, and he's not going to be happy. And I don't think he's going to survive that well. So we have to think about those little things. So it is an incremental thing that we work on, and it all depends on your resources. It depends on how much time you have as well. But don't give up. We just have to continue, because they are the driving force of everything. All right. Besides using a microscope, what other methods are there to know which compost is good or not? Oh, this is great.
What's your budget? Should be the first question.
Well, yeah, I think, how do we know what is a good compost? I think Dr. Elaine talks really beautifully about this, right? About the color, because we are a wonderful tool. We just stop trusting our gut instincts, we're just like, I have to use other things, but we're looking for that 70% dark chocolate color. We're looking for crumbly texture, certain moisture, good smells, earthy. So that's going to be like your high thing. But, I had some occasions, and I think I even shared one of those with Gerald in Cyprus, that we saw a compost and we're like, we're not going to have anything in this. This was like a 4-year-old compost. It looked dry, but we put under the microscope and it was so amazing. So I think we have some visual parameters that we can take a look at, but definitely the microscope is a great tool. I don't know if you guys have anything else to add.
Jean just reminded us of Dr. Elaine says that if you don't have a microscope, just get to know someone that does. So yes, Jean, that's another good path as well. But we know that especially when you start to work with large scale farmers, they want more. They want their mind is still wired on reports, tests, and those things. So if this is the case, there are some genetic tests that you can check to see which biology you have present. There are other groups investing on the genetic quantification of the biomass as well. But the system we do here that Dr. Lane developed over the 4 decades is just like easy, fast, and cheap. And like Isadora said, nothing better than your own system. So train yourself at the same time you are training yourself in the basic microscope, retrain your nose, your hands, your eyes to really feel the compost and trust the system. That will work black and white. And if your client wants the test, just give them the budget and go for. But you know for a fact that you can do the quick check in any compost.
That's right. Anything to add, Gerald? How do we know a good
No,
compost?
I feel like the first two of us are almost answering the
I know,
questions.
right? Everything.
Uh,
We just repeat.
I mean, there's different methods and different tools. If you have access to them, my thinking is the more evidence that you have of something being a good compost, the better, but it also requires some budget, right? So I think the microscope is a really powerful tool. One of the things that convinced me that this is a really good approach for soil regeneration is when I was able to see local commercial compost versus soil food compost that I was making. I could clearly see the difference. But there is other ways that you can see a difference as well. If you evaluate it visually, even if you smell it, you're going to get an idea about how good of a compost it is. Actually, if you investigate about the recipe, you can also get a pretty good idea of whether that's going to work or not.
Yeah, I remember someone told me, go to that place. They have really good compost. And I went there. It was a nice place. You can put your own shovel. I put it in a little bag. I closed it. I put it in my car and I went to pay. And while I was in line, an old man comes to me and says, oh, this is really good compost, but don't put it directly to your plants because you might burn it. I'm like, okay. Then I got into my car and I had a 7-minute drive to my house and my eyes were crying because of the smell of the ammonia. So
Whoa.
Yeah.
I think those are the signs that we perhaps need to stay away from. It's not going to give that power that Gerald was talking before. We're looking for a carrier for microbes here. This is
Yeah.
our probiotic, right? We're looking to put organisms back in soil, and the only way to do this is if we treat it as the respect they deserve, putting the good quality materials, understanding their process, and getting everything done. So yeah, so I think it's more
Yeah.
what's not good.
Yeah, Isa, I fully agree. I heard the same about the compost here. I had a carrot grower who asked me, about which of these two composts is better to use. This one has been recommended to me because I've heard that the chemical profile is very good, this and that. The only thing that they told me is to not use more than 30 sacks per hectare because it's so powerful that it can burn my plants. And then, I check it out under the microscope, nothing, no aggregates,
No
no
biology.
beneficial biology, just bacteria. Uh, it smelled pretty bad, like ammonia. So that's why I say, once you start making compost, evaluating it with your microscope, you can even eyeball if a compost is good or not. Sometimes we get those cool surprises like the one we got in Cyprus. But I mean, in general, you can tell the difference between something that is going to be beneficial and something that is not.
Yeah, and this is one thing that you can do to harness your balance as well. It's time to time just go visit places, spend some time there, like trying to sense the compost, check the texture when they're open. When it's in the bags, it's kind of difficult, but when you are in large areas, just spend some time there checking the differences, using your senses, and ask for samples to take back home and look in the microscope. A lot of times you don't even need to do the main assessment, like Gerald said, it will be bacteria, if there is bacteria at all. Because especially when you start to have compost shipping everywhere, big brands are sterilizing the compost because of the fear of spreading biology everywhere. So really look for the information on the location, their target market, what they are promising, and check under the microscope, test them. There is no better way.
That's true. I think maybe this is a question for you, Carla, because you mentioned this, but we can all answer anyway. They're wondering if you could repeat again why the bacteria under the microscope, that beautiful image that we had, indicated that soils were going towards an anaerobic state.
Yeah, we know a bit about that sample was from long time tea in protozoa infusion over a long time was overfed. So we had a lot of conditions in the system contributing to take out the oxygenation of the system. When you put a lot of food in a protozoa infusion, the aeration system might not be enough to keep the water in the levels that you need. And as the bacteria start to increase in numbers, they will consume the oxygen faster than the system can replace. And by the shape of that, it was a long bacilli, and the behavior in chains and how they're moving, we know that that's a behavior of type of lactobacillus. And lactobacillus, by effect, they thrive in lower oxygen conditions. So this is why we can, and like I said, based on that field of view only, if that is replicated in majority of field of views over that assessment, I would say the system is going to an aerobic state. So it's not just one field of view only. You really need to see the sample as a whole.
That's right. Anything to add, Gerald? Okay, let's move on then. And which lovely question. I love those. So would Bacillus bacteria be good? Because sometimes I feel like I think Soil Food Web, we try to simplify information, right? And sometimes things get a little bit, out of context too. And Bacillus, just like Dr. Carla was telling, sorry.
It's okay. Don't worry.
Just respect and utmost,
I appreciate that. Don't stress out. Thank you.
Just, Bacillus bacteria, like Carla said, they're facultative bacteria. So it means that they can actually work under two different sets. They have two sets of enzymes, and they're okay with working with a lot of oxygen and maybe a little bit less too. They will work around that. So they're not necessarily an excellent sign for aerobic conditions, but my take is that there is a niche for everything. Bacillus are very, very powerful bacteria. They are wonderful. They have many, many functions. The soil must have microaggregates with little anaerobic pockets as well, but it's just about the amount, right? We don't want a soup of it. We want just enough so they can do their job and they contribute to the system. But yeah, and not, we're not adding straight yeah, lactobacillus like soups to it. But yeah, I think that's my take on it. I don't know if you guys want to add anything else.
And it's about perspective, right? Because lactobacillus in soil, they indicate a not so good condition for the plants, but inside our bodies are the ones that we want, the majority of species. So again, we have to go back. This is the point where it's beneficial to try to dig in into the species level. When you talk about bacillus with the big B, we are talking about a genus, which has tons of different species on it. So which one you're talking about? If you have bacillus with small b, we are talking about the morphology of a group of bacteria. And then we expand even more. We can have aerobic, facultative, anaerobic bacteria, but also have to understand that being anaerobic is not similar to the disease causers. Same way for aerobic organisms. So it's very important question. Thank you, Jose. Allowed us to expand a little bit on how we need to be careful when we put labels in specific groups saying, oh, if it's a bacillus, it's an aerobic disease causer. No, be very careful. There is all the information we need to gather before putting these labels like disease causers, for example. What else, G?
Uh, well, it's related to what I was mentioning before. Our goal is to have a diversity of microorganisms. I like when Isa says that we don't have to micromanage them because actually we are still in the process of understanding many of those, right? So why micromanage them? This is a reason why our approach is enhancing diversity and the conditions, because that's what a forest does. It just keeps the conditions, and the right species will come. Plants will manage that. So that's one thing that I wanted to mention. The other thing is there is never a total absence of inoculum in a system. There is even pathogens or biological control agents in a healthy soil. What we want to have is a balance of biology and the conditions. If a specific species of bacillus is important at some point during the transition, why not use it? You just have to be aware of what is the effect of that, and you want to be mindful of not causing an imbalance in the system. That's what I would add to what Carla and Isa brilliantly said already.
Some wine growers, they like to use lactobacillus as leaf, as foliar, because they will do a very good control agent, and they're not going to thrive there, all of them. They're just going to create a very nice biofilm. They might even create products that will, avoid others or consume the food sources in the phytosphere. And so they could be a useful tool, but you just have to understand the context of everything, just like Carla and Gerald said. Yes.
Beautiful.
Awesome. I'm living in Southeast Asia, where it's hot, warm, and humid. Are you sure you're there? How do I identify if the white stuff that we saw are good mycelium fungi growing or it's bad fluffy mildew? That's so nice. We were now in the UK, and that humidity there is kind of crazy for me too. And some of our compost had very nice fluffy stuff on top as well, I mean, thinking that mycelium takes time to grow, right? So we're still going through the thermophilic phase. I'm just wondering, okay, so I got some of it and actually put it under the microscope, and it was pretty awesome. We had
I need the
naked amoeba.
pictures.
I don't know if I took pictures because I went rogue. Everyone was talking with their piles, and I just grabbed thing and I just went to the microscope to check it out, and it was so cool. I saw naked amoebas, even nematodes. It was a really different environment that I'm used to. But yeah, I don't think we should be so scared, especially when we are in the early stages of decomposition about anything being, just understand what's telling you. Perhaps you have an excess of moisture. Perhaps you have the right conditions for those decomposers to start working under those circumstances, and maybe try to balance it out, and okay, so how can I get some airflow in here or something like this, just to make sure that we even the game, right? We even the rules here of the game.
And when turning the pile, put that portion in the hot center to control again by the temperature. And now we have, this is the webinar of nicknames. Now we're gonna call you Rogue Isa. Sorry.
Like that. I feel like I'm an Avenger now.
But I agree with you. Every time you have a chance, just get the sample under the microscope. If you cannot do that, let's say you are turning the pile, take a sample, put it on the side, do your work, and then as soon as you can, go to the microscope and try to associate what you're seeing in the microscope with the visual cues you have in the pile. So for the next ones, you don't even need to go to the microscope again. This is how you're going to build your mental microbe library. And as you get more of those experiences, faster your decisions will appear. So it's just like really open up your mind to allow yourself to make these external connections, what you see in the field, what you see in the microscope. Talk with other people that are nearby. Have you seen this before? You might get extra tips of how to control this outbreak because someone already passed through that. And again, the forum is a huge ally. We have amazing students sharing their experiences and how they are dealing with things in different parts of the planet.
I wanted to add something here. If that portion of the compost already went through a heating cycle, I wouldn't be too concerned about it, and I would take it more, as Isa said, as an indicator. Maybe something is causing an excess of moisture that is lowering the oxygen conditions a bit, and that fluffy stuff is finding an environment for growing. Also consider that mildew is an obligate parasite, meaning that it cannot feed on decaying organic matter. And many of the oomycetes are organic matter decomposers. So it's also, if you're concerned about a specific type of, a pathogen or something, getting an idea about the ecology of it will also help. In this case, it won't be a possibility for mildew to survive in a compost because it's not saprophytic, it's an obligate parasite.
There you go. Yes. Thank you, Gerald. It's true for that. We have 8 more minutes, so I don't think we have time for a lot of questions anymore, or else we'll just go super fast.
Let's do our best.
What can you do if you have root-feeding nematodes? My compost got too wet and has not really recovered. I added wood chips and I have aerated regularly, but it's not smelling good and it's too moist. Is this two different questions? Or is it
Almost there,
in
yeah.
the compost? I don't know.
Okay.
What can we do about root-feeding nematodes? I'm assuming this is in soil, or it's not in compost, right? Because compost doesn't have any roots. It's not so easy to find root-feeding nematodes in the compost. Although I have seen, but not in a lot of features.
Yeah, my favorite approach on that in the soil is to get a good compost where I have a lot of predatory nematodes and make a good nematode extraction and apply in the areas that I'm seeing the root-feeding nematodes. Also check what else is affecting the plant, which are the other missing organisms in the soil that is not giving the plant everything she needs to grow healthier and giving the predatory nematodes time to really chop down the root-feeding nematode population. So I see this as a multiple facets approach. You have to bring back or boost up the predatory population at the same time, give the plant a chance to recover while you are dealing with the root-feeding nematodes. Sometimes if the outbreak is too intense, using alternative of biological control that might not be so specialized, that can be like a general nematode killer, but sometimes it's better to just cut the bed on the root, like taking out of the root-feeding nematodes, and later bring back the beneficial nematodes to the system because the damage might be smaller in your yield. So really depends on the level of fruit-feeding nematodes you have in the area. Do you want to cover the compost too wet, Gerald?
Do you ever get too wet compost in your area, Gerald?
It's Costa Rica.
Yeah, I really,
we had conversations about this.
yeah, I really try to avoid that. It can be, yeah, a headache. Um, well, I think it really depends on how wet the compost got. Because there is a point that is very difficult to get rid of the moisture. Um, but what you're doing is already a positive thing. Adding wood chips to aerate. Just be aware that if it is thermophilic composting, you want to add those wood chips at a certain part of the process. I would say that's an option. Also, yeah, maybe, I don't know. I don't have many background about this, but if it is already maturing, you could even add worms. You could even punch a couple of chimneys to help aerate the compost and help release some of the moisture. And that way you will eventually get to a better temperature range. No, sorry, a better moisture range. That's what I would recommend here.
And there is the other scenario as well that sometimes doesn't worth all this extra work. Just deassemble the pile, let it dry, get back to the 50% moisture, and recompost that in another thermophilic pile. Sometimes you have to counterbalance, do I have the time and the bandwidth to try to recover this pile, or at this point it's better to just recompose that again. There is no losing material here. You just need to decide what is better for the condition you have.
Exactly. Karen, you asked about chimneys. Yeah, it's basically punching holes in the compost.
Yes, someone in the chat was asking. I think we're gonna wrap it up. I feel kind of bad. I would sit here for hours with you guys, and thank you for everyone who came. I just want to give a really quick reminder that this is not the end. We have 3 more parts to go. Next week, we'll be with Nick, with Brian, Dora, and myself, and we'll be talking about that kind of compost. How do we actually multiply those organisms? And then we're gonna actually see the work that those organisms do on webinar number 3. And then we can all chat together and get to meet each other a little bit more in webinar number 4. So for now, I really want to thank everyone from the Soil Food Web that was here working today. Carla, Gerald, it's such a pleasure to be here with you guys. And everyone else who was here with those excellent, great questions. I hope we inspired you guys a little bit more. I hope, yeah, we answered more questions that we've given you, because I know sometimes we just like, we just give more questions, but yeah. So do you guys want to say anything?
This is how we evolve, right? When you answer one question and the other side minds just pop 5 more. So this means that we are really staggering what you have to do. I had a lot of fun. Thank you so much for another great webinar. You guys make our day every single time. I cannot wait for the next one. And for the community house party and for the other webinars as well. But please come with open heart. And on the webinar, on the house party, remember that we will be there, listen to you guys. So it's your time to do the revenge. Just talk nonstop. We're gonna be there having fun and learning from you. We love that time. Thank you so much for this incredible webinar. Gerald?
Thank you very much for making me part of this. I'm really grateful to have shared this time with all of you. I'm very happy to see people from different parts of the world. It really fills my heart to see people who are looking for better ways of doing agriculture and restoring our systems. I'm also very happy to share with Carla and Isa. You both are great. It's really easy to help people, with their questions when we have this kind of support. I love the Q&A, and I could go forever with this. But yeah, well, looking forward to being in another webinar in the future. Thank you all for your time and for being here, and hopefully we cross paths at some point in the future to continue contributing to this great cause.
All right, so yes, I see you guys next week, 17th.
Bye-bye.
Bye.